RNA processing occurs in eukaryotic cells during the end of transcription. The pre-mRNA formed during the stages of transcription must be edited through RNA processing before the code it carries can be used in translation to synthesize proteins. There are 3 steps to mRNA processing. First, a modified guanine cap is added to the 5' end of the pre-mRNA strand. Then a poly-A tail is added to the 3' end of the strand. And finally, introns are removed through splicing and the remaining exons are ligased back together. The cap added to the 5' end of the pre-mRNA strand is a guanosine molecule with a methyl group attached to the 7th member of the guanine ring system. The cap protects the mRNA strand from being broken down in the cytoplasm and also assists the ribosome attaching to the strand during transcription. The polyadenine tail consists of repeating sequence of adenine bases. The pre-mRNA is cleaved at a cleavage point along this strand. The signal for the cleavage point is AAU, AAA, which is located 20 to 30 bases upstream of the cleavage site. In around 70% of mammalian pre-mRNA strands, there is a second signal present, a section with a high proportion of G and U bases, around 20 to 40 bases downstream. The AAU-AAA binding protein, an endonuclease, and the enzyme poly-A polymerase then facilitate the addition of the poly-A tail to the cleavage site. The poly-A tail makes the strand more stable, increases the efficiency at which it is transcribed, and helps the mRNA to move from the nucleus to the ribosome. Eukaryotic genes also contain stretches of DNA which do not code for polypeptides, called introns. Here they are labeled in red. They must be removed before transcription can occur. Introns vary in size from 200 bases to 50,000 bases long, and they are more prevalent in higher eukaryotes such as humans. 90% of our genes are interrupted by introns. The areas of DNA that do carry a code are called exons, which are highlighted in green here. Introns are removed through splicing. Introns start with GU and end with an AG, and they are identified by spliceosomes, which are complexes formed from small nuclear ribonucleic protein particles, for short SN or NPs, and proteins. The spliceosome catalyzes the extraction of the introns from the exons. After the extraction of the introns, the exons are ligated together, and the process is completed. The mRNA is now ready to move to the ribosome. The pre-mRNA is now mRNA and is ready to move to a ribosome for translation.